Welcome to part 2 of the webinar series about on-farm variety trials to manage risk for organic and specialty crop producers. The two webinars in this series are part of an on-farm variety trial toolkit, which you can find at the link on your screen. This is your host, Alice Formiga of eOrganic. eOrganic has many articles, videos, and webinars about organic farming and research, and you can find our resources on our website at extension.org/organic_production, as well as on the eOrganic YouTube channel. In this webinar, you'll be learning about how to evaluate and analyze your variety trials. I'm very pleased to welcome back our presenters today, Julie Dawson of the University of Wisconsin-Madison and Kit Healy and Jared Zystro of the Organic Seed Alliance. They'll be speaking for about 45 minutes, and then we'll have 30 minutes for questions, which we'll be reading out loud after the presentation is over. Dr. Julie Dawson is an assistant professor for urban and regional food systems in the Department of Horticulture at the University of Wisconsin-Madison. Her research focuses on variety trials and breeding for organic systems in the upper Midwest. Kit Healy is the Organic Seed Alliance Research and Education Associate for the Midwest region. Her master's research focused on conducting tomato variety trials for short-season organic production and engaging chefs and local farmers in participatory breeding and evaluation projects. Jared Zystro is the Research and Education Assistance Director at the Organic Seed Alliance. He manages organic seed system development in California, conducts participatory breeding projects and variety trials, and teaches farmers about seed production at workshops and field days. So now I'm going to move on to the main presentation and pass the screen control over to our first presenter.
Thanks everyone for joining us again for our second part of this webinar on trial evaluation and analysis. Just before we start, Alice mentioned briefly that this is part of a bigger project that is included in an online toolkit hosted by eOrganic. And so just before we jump into it, I just want to mention a few other parts of this project, which is generously funded by the USDA Risk Management Agency. I forgot to put their logo up. It's my bad. Don't tell anybody. So the RMA is a partner on this project. Moses, the Midwest Organic and Sustainable Education Services, are a partner. And then Organic Seed Alliance and UW-Madison are bringing this all together. And the parts of the project that are additionally available to you all to continue to advance your skills in variety trialing are the Grower's Guide to Conducting On-Farm Variety Trials, which is available online, the recordings of this webinar, as Alice mentioned, and then this summer we're hosting 4 on-farm workshops throughout the Midwest. 2 of those are in Wisconsin, in Westboro and Janesville at We Grow LLC and at the Hughes Farm. And then we also have sites in Illinois and Minnesota. So if you are proximal to any of those locations, it would be great to see you out at a field day where we can actually walk you through an on-farm variety trial, talk to the farmer about why they decided to do a variety trial and what information they're interested in getting out of it, and then actually see how the trial is laid out in the field and talk through all of the different pieces of conducting, evaluating, and analyzing the trial. So we hope to see some of you out at those events this summer. So just to review quickly, last time we focused on how to set up why you might want to do a variety trial, and then how to set up the trial and how to do the trial design. So one of the 3 important pieces that we talked about are defining your trial goals, so trying to be as specific as possible about exactly what information you're hoping to get out of doing a variety trial. That could be any number of different things, like you might be interested in looking at something like yield, where you want to compare different varieties to each other and how well they yield. Looking at flavor is another thing that people trial for. Specific disease resistances, early vigor, or for a specific planting window. So maybe you have something that does pretty well early in the season, but you need something that performs excellently later in the season. A trial for a specific planting window is another goal that you could set, or maybe to fill a particular market gap. We also talked about the difference between screening and replicated trials. So screening trials being a type of trial where the varieties are each replicated one time and then compared to a common check variety that's replicated multiple times in the field. And the idea with a screening trial is that you could look at a larger number of varieties without maybe taking as detailed of information about each one as you might with a replicated trial. So screening trials are better for an initial assessment of how varieties do based on your trial goals and what you're interested in. And then you might want to follow up with a replicated trial in which each variety is planted in at least 2 plots across the field. And this helps to account for environmental variation and get more reliable results. With a replicated trial, they do tend to require a bit more work. So you have to be sure that you have more time to observe each replication so that you're not shorting yourself on getting the reliable results that you're interested in. We also talked about trial design, mostly focused on the 2 main points of minimizing the effect of field variation. So when you're doing a trial, you're mostly interested in seeing different varieties perform based on the varieties themselves, not based on the fact that one plot was planted in a wet part of the field and another plot was planted in a dry part of the field, and that's skewing your results. So you might include border rows, avoid field edges, and avoid known problem areas in the field to minimize the effects of field variation. And then the second important point for trial design is to be sure that your trial is integrated into your farm operations so that you don't have an additional piece of work that you're not accustomed to thinking about. So if your trial is kind of at a strange part of the farm, or you're trying to do it at a strange part of the year that doesn't flow with your general farm operations, or if you design a trial that's just too big, that's taking on too much, or asking questions that are too difficult to measure, you're much less likely to follow through. And the idea is to do something that is reasonable to follow through on. So that's kind of where we left it last time. Let's see if I can advance the slide here. There we go. And we looked at Jared introduced to us 2 real world examples of variety trials that he's been a part of. The first one was and we're going to review these now just very quickly because we're going to come back to them as we discuss trial evaluation and data analysis. So the first trial that we talked about was a green bean trial at Deep Seeded Farm in Arcata, California. The goals that were stated and stated very clearly and specifically where they were looking for straight pods, white mold resistance, flavor, ease of harvest, and the length of the harvest window. So those were the specific measurements that they were interested in taking. And then the design that made the most sense for this farm was to do a replicated trial that had 2 blocks. So you could replicate many different times depending on how much space and how much time you have. But a lot of times people just pick 2 blocks to cover the environmental variation. And so that means that each variety was replicated once within each block, and then there were check varieties also across the blocks, and they looked at 15 trial varieties. The second trial example that we talked about was a spring wheat trial at the College of the Redwoods, also in California. The goals for this and a grain trial often is a little bit different, and we'll be able to talk about that more on this section of the webinar as well. They were looking for high yield in their organic system, which was also a coastal organic system, so a unique environment. Looking for stripe rust resistance, weed competition, and quality of the grain that was produced. And the design that made the most sense for this trial, I remember Jared put up a trial design graphic that we could all look at if folks are interested. So that was also a replicated trial, but it was in 2 different locations. So not just multiple blocks in the same field, but actually on 2 different farm sites. And then there were 4 blocks on each and 8 different varieties evaluated within that trial. So those are just 2 examples of replicated trials. And we have many more examples that we can also talk about during question and answer if folks are interested in something more specific. So now I'll turn it over to Julie for some more specifics on how to evaluate your variety trial.
Great, thanks. So in evaluating the trial, it's important to think about the goals of the trial that you laid out at the beginning, what's most important for you to get information on, and also being realistic about your time commitment. So the traits that are important you think about when you're designing the trial, we talked about that last time. Those traits will usually have an ideal time of evaluation, and you have to kind of assess if you're going to have time to evaluate the trial when it's ready. And then today we're going to talk a lot about how to record those observations and tools that you can use for data collection and analysis. So for evaluating, usually you want to make a list of the traits that are the most important. And one of the things that we try to do in trials is have the same person take a particular trait. So it doesn't have to be the same person taking all of the data on the trial, but if you're taking disease measurements through the season and saying you want to rate early disease incidence on your trial and late disease incidence on your trial, you want the same person to do that for all the varieties so that you're sure the measurement is consistent and you can compare varieties to each other and you don't have variation due to the person that's doing the evaluation. So try to have one person take notes or data on a particular trait throughout the season. We're going to go through a few different ways of measuring trials. A lot of these strategies are described in more detail in the on-farm variety trial guide, so you can download that if you want a lot of detail. We're going to go through them fairly quickly today, and then you can look it up in the bulletin, or feel free to email any of us if you have questions specifically on different strategies. So the 4 strategies we're going to talk about is actually measuring the trait, scoring, ranking, and qualitative notes. So measuring characteristics is when you take an actual measurement, whether it's a length or a weight, a height. This is the most precise way of measuring things usually, but it also can be time consuming. So you want to ask yourself whether you need a very precise measurement to assess that trait, or whether you could do something that's more rapid, like a score or a rank, and get the same information for decisions. Measuring characteristics can sometimes be faster than scoring. If scoring takes you a lot of thought to decide whether something is high or low. In the middle, sometimes it's difficult to score something accurately. So sometimes a measurement like a plant height actually goes faster, but usually this will take a little more time and is useful for traits where you want a pretty accurate measurement. So you might decide that you need an accurate measurement of yield, so you're going to weigh things coming off the field. Or you might decide that, just a comparison of the varieties where you know which one's the highest yield and which one's the lowest is fine with you. So that's measurement. You're actually taking a measurement. Scoring is when you have a 1 to 9 or 1 to 5 scale. 1 to 9 gives you a little more ability to differentiate things if you use the full scale. So usually 1 is poorest and 9 is the best. You use the odd numbers as the primary scores, and then even numbers for varieties that are a little bit in between. Sorry, that cut off. The text got cut off on the slide. So first, when you do a scoring trial, since you're not actually getting a measurement and you have to judge how varieties are performing on this scale, the best thing to do is to walk the whole trial first and decide which one is the worst, which one is the best, find one in the middle. So you have an example of a 1, an example of a 5, an example of a 9. It may be that a lot of the trial looks really good or a lot of the trial looks really bad, but you want to actually use the whole scale because the purpose of the trial is to try to differentiate varieties. So if you score everything 4 or 5, you really don't get that much information out of it because everything looks the same at the end. So you might do a score for, say, disease susceptibility or resistance. you would have 9 be a perfectly healthy plant, 1 be a dead plant, 5 be something that's partially affected, and then kind of set the trial by looking for the healthiest plant, the worst plant or plot, and then kind of looking for one in the middle, deciding that's 1, 5, and 9, and then you can go through and score each plot for the disease incidence. A similar but slightly different way of evaluating trial is a ranking. This is where you force yourself to rank the varieties from best to worst. With a score, you could give more than one variety, more than one plot the same score if you thought they were truly the same. With a rank, you try to force yourself to actually rank it from best to worst relative to other varieties in the trial. So with this, you would use a similar system where you would walk through the trial and decide the best and the worst, and then try to rank them in between. This is an example of a ranking where you are looking at appearance, flavor, and the marketable fruit. Marketable fruit, you could use a measurement, and this looks like maybe they did use a measurement in pounds there, but you could also use a ranking where you maybe harvest everything in front of the plot, and then you walk through and you see which one's the best, which one's the worst, and that will set your rank number 1 as the best, that's the highest yielding, and then the worst, and kind of line up things in between. This can be faster because you're really focusing on comparisons between the different varieties. But sometimes if it's like yield and it is difficult to decide on the ranks, you could just weigh it, and then you know you have a pretty precise measurement. The last strategy is qualitative. This is where you're focused more on taking notes and on recording your impressions of the plot. This can be really important in addition to the other measurements, so you should always take notes on the plot so you can remember what they look like, what you were thinking at the time about how they compared to other varieties. It's hard to remember when you're not in front of a plot what it actually looked like, and so writing down your impressions can be really important to interpreting the rest of the data if you're using measurements or rankings or scoring. You can also do a trial where you just take qualitative notes. In a screening trial, when you're trying to decide which ones to continue, you might just write down what are the good traits and what are the bad traits about particular varieties, and then they might go into a more detailed trial the next year. You do want to be careful not to say the same thing all the time. So if you're scoring something for flavor or for disease susceptibility, if you say everything is pretty good, by the end you're not going to know which one was actually better than the other. So try to use some detail when you're taking notes, and try to be fairly systematic about it. If you're going to take notes on something, walk through the whole trial and make sure that you have all of the plots accounted for, so that you know that if you like a certain variety and it doesn't have any notes about disease incidence, it's not just because you forgot to take notes on disease incidence for that plot, but it's really because it didn't have any diseases that were significant. So in general, for data collection, you want to start by walking through the whole trial, evaluating what it looks like overall, what the good plots and the bad plots are, and then evaluating each plot individually for one trait at a time. It may seem like it's faster to do all the traits on one plot and then all the traits on the next plot, but usually you are better at comparing things when you're doing one trait at a time. You want to look at the whole plot. Don't just focus on a good plant or a bad plant, but really evaluate the plot as a whole. And then whatever strategy you're using, it's often good to, at the end, really summarize your impressions and rank each variety for all of the traits. So the best variety overall and the worst variety overall, that can help you in interpreting some of the data as well. And make sure that you keep your data sheet safe, keep it in your computer, or make more than one copy. Don't have one copy that if something gets spilled on it, you lose a lot of work. So make sure you label the data sheet, have the plot numbers and the variety names, and then keep it safe. I wanted to spend a couple minutes talking about taste tests because this is something we get a lot of questions on. To do a good taste test is not complicated. You just need to remember a few things that make it easier to compare varieties to each other, and also get some descriptions that you might use in either a farmer's market sale or in describing it to another farmer. You want to make sure to taste the crops when they're mature and to try to get everything at an equal stage of ripeness, so that you're comparing varieties at the same stage of ripeness and you don't have one that's different than the others because it's a little underripe or a little overripe. Be aware of what's happening in the environment. So tomatoes, for example, will taste different after getting 4 inches of rain than after a week of no rain. So make sure that you're aware of that. Get a representative sample when you're tasting a plot. Try to get more than one fruit so that you have an idea of whether there's any variation within that variety. Use common sense with food safety. Don't leave things sitting out for a long time after they've been cut up. Make sure you have the ability to wash things if necessary. And don't talk to other people that are tasting until you're done, because you can really influence other people by saying that, oh, that's the best, or yuck, that wasn't good. Certainly influence other people's opinions. So don't talk until the tasting is all done. When you're doing taste testing, you really have two potential purposes. One is, do I want to keep growing this variety? Does it taste good or not? Should I keep it or should I drop it? And the second is to be able to describe some of the flavor. So with deciding whether you keep it or drop it, it can be an overall preference rating. You don't have to have a lot of detail, but you do need to know whether people like it or not. So it could be a 1 to 9 scale. 1 is poor and 9 is excellent, or you could just do keep or drop, so kind of a binary scale. You can do more varieties with this if you get going and really focus on it, but you do end up getting some taster fatigue after a while, so you want to be careful about how many varieties you try to taste. For description, you want to get some information on different aspects of flavor, so you usually are focused on fewer varieties, but try to have more tasters. And you often use a scale rating. So this is the scale that I talked about before, only we use a 1 to 5 for flavor. You could use a 1 to 9 if you wanted. We usually have some things that are aspects of quality that aren't really flavor, so appearance and texture are the big ones. And then flavor components like how intense is the flavor, sweetness, acidity. We might add 1 or 2 depending on the crop species. So for hot peppers, we have a spiciness rating. For brassicas, we have a bitterness rating. For beets, we have earthiness. So you can add specific ones depending on what crop you're focused on. Sweetness and acidity usually are important for most of the fruits and vegetables that we work with, and intensity tends to be correlated with preference for most crops, although sometimes like summer kale, if it's really intense and bitter, people don't like it. In terms of practicalities, what we do is we take a representative sample from the plot, we cut it up, put it in a container, and label it with a 3-letter code. We try to focus on 6 varieties or fewer, and we provide people with water and unsalted crackers if they need a palate cleanser in between. We try to have people taste samples in different orders so that not everybody will taste the first sample, TRM in this case, and then ZCL and then ARX, because that can influence how people experience it. The first sample, if you're hungry, will taste better than the last sample. So you want to try to get people to taste in different orders, and you want to keep the rating sheet as simple as possible. So for having multiple flavor components, it could look like the one on the screen. If you're doing a preference tasting where it's just 1 to 9, you might have each variety and just 1 score, 1 to 9 or keep or drop. You want an area where people can write comments if necessary, if something is off, or if they really like a variety, have a place for them to put that so they don't put it in one of the other boxes. And then you can use the methods of analysis that Jared is going to talk about to analyze this kind of data from a score. You can also do a qualitative evaluation. We found that sometimes it's hard to find descriptors for varieties, so it's hard to come up with what they taste like. And we're working some with the Culinary Breeding Network out in Oregon to develop some lexicons for varieties that might help with tastings. So with that, I will turn it over to Jared, and he will talk about analysis, what to do after you've actually collected all your data.
All right. Thank you, Julie. Mentioned that the analysis side of it can be, it's sort of the final hurdle. You've put all of this effort into conducting a variety trial. You've gone out there, taken space in your field to plant this, had to manage multiple different varieties, which is usually a larger burden than just growing a single crop. You've had to collect all of this data, and now how do you make use of this data to make good decisions? So a lot of that comes down to, are there effective ways to kind of summarize the information, make it less overwhelming, get to the truths within it? And there's often a balancing act between being able to see the overall picture and then being able to look at the details to get answers to some of your questions. So going back to all the way back to your trial goals, it's always good to reflect on what those goals are when you're starting to analyze your data, to get a sense of how much do you need to go into what specific traits. Are you looking for kind of the basic, just, is this a variety I'm adding to my farm? Is this a variety I'm dropping? Which of the traits are make or break traits, where if it's not at a certain level, or it's not the best for this trait, then I'm not interested in it. And which traits maybe are just gravy, where you're interested and would like to find something that's superior for a given trait, but it's not as important. So kind of going into your analysis with some of those questions and priorities in mind really help. So with that being said, let's take a look at what you're often starting with. Data that you collected that is this, one or more of the different ways that Julie talked about, how data might be measured, whether it's, actual measurements or scores or ranks, whether you've collected it in a screen trial, whether you have, multiple replications, but often you're looking at numbers on a spreadsheet along with your notes and your photographs and trying to summarize all of that. So let's take a look at some of the tools with a focus on kind of the numbers here. So let's use this as an example. You may have a set of numbers, spreadsheet looking at recording all of the scores that you gave for wheat rust resistance in your trial, and say that you had 3 replications where you planted each of your varieties 3 times in the trial, and you walked through the field and you gave each of them a rating, with 1 in example being the worst and 9 being the best. So if you look across this, one of the simplest ways to summarize and simplify your data for analysis is just taking an average. So if you just do an average across the 3 times in the field that each of these varieties were planted, you get a sense of overall how much rust resistance was I observing in this trial. And that will give you a sense in turn of kind of the, you could put these in order and say, okay, looks like, Lassic came out on top. Then after that was Hollis. Then after that, Sonora, then Red Fife, then Foisey. So that is a pretty straightforward way to get a sense of your winners and losers in the trial. However, one of the things that can be important to keep an eye on in trials is, especially one of the strengths of replication, is that you're able to observe the variety in more than one place in the field, and it gives you a sense of uh, whether or not there might be either field effects or some degree of uncertainty in the way that you measured things, or in the amount of variability in terms of how that crop grows. So taking advantage of that, you can actually look at the individual reps and start to ask yourself a question. So something that pops out here is that if you look at Hollis actually in rep 2 was better than last. And in rep 1, it was just about the same. It was a 7 versus an 8. Where it really fell down for some reason was in rep 3. And so why did it rate a 3 for what rest resistance in rep 3 compared to the other reps? What was going on there? You'd expect that if this was true resistance in Hollis, it would have consistently good ratings. Was there something where maybe Hollis just was lucky in the other replications and kind of avoided some rust pressure? Was there a gradient going on where you start to notice that rust seemed to be more present on one side of the field than another? So looking at something an outlier or something strange where one of the varieties or one of the replications for variety performed differently than the others, let's have an opportunity to have a little more questions about what's exactly going on in the trial. The other thing that you can do is you get a sense of just how much variability there is for a trait, and how that gives you a certain degree of humility in terms of the level of certainty that you might have in your ratings and in your rankings and in your decisions. So that uncertainty can be looked at more formally using some statistics, and that's what I'm going to show you now. I'm going to show you a tool that we developed for farmers and researchers who want to be able to design and analyze their variety trials. So this is our variety trial tool. It's at seedalliance.org/variety trial tool. And right now it is in beta. So there are still some improvements that we want to make to it, but it is currently usable for anyone that wants to make use of it. And basically the way that it operates, if you look at the top of the screen there, it goes 1, 2, 3, 4, 5. You kind of step through it from initially using it to help you plan your trial, talk about some of the things that we talked about in this webinar in terms of creating, you can either create a randomized complete block design where you have full replications of each variety. Each variety is replicated multiple times, or what we are calling an augmented design, where you have just a handful of the varieties to serve as checks that are replicated multiple times, with the rest being unreplicated. So you can design either of those using this, and then enter in the entry names, how many replications you want, and the traits that you want to measure, and it will make a little spreadsheet for you, as well as a map if you want it to make one. And so that can serve as your data sheet, which in turn will allow you to have your data in a format that allows you to analyze it using this tool. So it'll look kind of like this, where you'll have plot numbers, though that's not strictly necessary. You'll have the replication, the entry in the replication, and then for each of those plots, you have different traits that you're measuring. So, in this case, this is a green bean trial at Deep Seeded Farm that already mentioned, where we measured weight, flavor, appearance, and texture of some of the measurements. And what you can see here was that in the first replication, we collected a complete data set, but then in the second replication, in part, I think what Julie said, where it's hard to get taste data for more than 6 varieties, the what we compromised on was to have a subset of the varieties that we tasted 2 times and looked at for quality twice, so that way we get some sense of how much variability there was, even if we didn't replicate the data collection everywhere. So that's sort of what you're seeing there. So now you have this big muddle of data, of numbers, and how do you make sense of that? How do you make decisions? Well, if you use the format that this tool provides in terms of the spreadsheet to collect your data, it should be relatively simple then at the end of the year to go to step number 3 here and upload your data. You just go to the browse button, select it, and upload it, and then it will get uploaded into the website, in which case you can go over to number 4 to analyze it. And so what this provides right now is 2 visualizations that you can use to kind of quickly get a handle on your data. Right now it does a plot of individual traits, give a sense of both the average for that trait and the amount of variability that exists in that trait, so that you can sense which ones are genuinely superior to each other. So, in this case, we're looking at the total weight trait. Let me zoom in a little here. And so you can see is that in this case, the black dots are the individual data points that were collected at each replication for each variety. The red dot is the average across those 2 replications, and then the red line is the amount of it's the confidence interval where we could be basically 95% sure that the true average of this variety for total weight would be somewhere within that range of that red line. So, in this case, you could OSU 5402 here, the highest rated thing is, basically, its average is higher. OSU 5630, they're very close in terms of, being within each other's ranges, whereas OSU 5402 is basically, outside the range, superior to just about everything else that we measured in terms of total weight. Oops. Whereas with flavor, in part because multiple measurements take a handful of varieties, we have more uncertainty in our measurements. So in this case, we can see, well, clearly, this even despite the level of Bronco, we rated, it rates more poorly than, the vast majority of things. And the OSU 5402, also is, though we can't be certain that it's better than some of these other high performers, we can feel pretty confident that it's better than, say, Jade or Fresh Pick or Bronco or Caprice. So that's looking at individual traits, but if we want to take a look across all the traits to get a sense of how did things perform overall, the other kind of analysis that this online trial tool provides currently is a summary table that's color-coded using a colorblind friendly color scheme. So the way that this works, and let me zoom in on this, if you select over on the left-hand side where it says means table and click analysis, it will produce a table that looks like this, that summarizes across all the different traits that you looked at. Let me zoom in on that. And so what you see is that on the left-hand side, it has all the entries that you looked at. And then the next thing you'll see is for the average of each of these traits across all the replications, and they're color-coded so that purple is a low number and 10 is, yellow is a high number. And so, it gives you kind of a quick eye towards it. The second thing that you'll see, the second column, say after total weight here, is you'll see a bunch of letters. What do those letters mean? Those are what are known as means groups. So this will give you a sense of if a variety shares a letter with another variety, then based on those confidence intervals, those red lines we saw, it's essentially statistically the same for that trait. So, in this case, you can see OSU 5402, is got a means group, is in means group A. That means that it is basically the same as, or we can't be confident that it's any different for total weight than fresh pick, OSU 5630, or pike. But we can be pretty confident that it's superior to the rest because it doesn't share the letter A with any of the others. And so going down the line, you can see the same kind of coding for flavor, appearance, and texture. What you can see here is for appearance and texture in particular, we didn't have enough, we didn't replicate enough to get to feel confident that these varieties were genuinely different. They all share the same means group. And so that statistics allows you to kind of have a degree of, like I said, humility to say, okay, we really not have enough information here to make a clear determination of which is better for those traits. So the other thing that you can do with this tool is if you don't want to look at this online or you want to save it for later, you can, as you get each table or graph, you can attach them to a report that you can download. So you just click, as you create a table or graph, just click attach the report, and then you can give a little bit of information about the report, like its name, location, description, and download PDF report, and it will download a PDF for you that has a title page and then has all of those graphs that you generated, so that way you can save it for later. At that point, the last part of this tool that I wanted to share with you, and encourage you to consider using this tool, and if you do, the final number 5 here on the top says save and share. So if you want to save your data set online to allow you and others to be able to view it later following these, what says that this data here is deposited in accordance with the Toronto Agreement, which means that if other people do look at your data, that if you're, say, a researcher and you want to make sure that you're the person that publishes information, that when you deposit your data here to share it with other people, it's saying that other people can't use your data to develop new publications, that you have the right to that. But if you do want to save and share it, you can just fill out a little bit of this information, and the email address is not public information. That's just something for me to be able to contact you if someone does want to use your data, and then hit save data, and then it will be available for others and for yourself to use later. So that was walking through the first example with the green beans, which were locations at one site. Now I want to walk through the spring wheat trial example, where we have more data. We have more traits, more varieties, more replicate. Well, actually, not more varieties, but more replication and 2 locations. So, all of a sudden, you're looking at a lot of data here, and how do you make sense of all of this data here? We collected rust data at College of the Redwoods. We didn't collect rust data over at Front Porch Farms because there wasn't enough rust pressure to see any differences. And we collected heights at College of the Redwoods and at Front Porch, collected yields at College of the Redwoods and Front Porch, and collected protein at College of the Redwoods and Front Porch. In fact, there's even more data than this. This is just to show today. So, can you make any sense of this? I can't just by glancing at that. So how do we summarize it in a way that we can make decisions? So right now this is set up in the same format as the green bean trial data that allows us to upload it into the variety trial analysis tool. So again, just go to number 3 here, upload your data, click on the CSV file. That's a comma separated values file. It's kind of like a stripped down Excel file that can be used to save your data, and that's the format of the data sheet when you download it from this tool as well. So as long as you use this tool to design it, or you can download it right where it says here, you can download the template for it. Click on one of those links that says download this template for RCBD. Then you can go ahead and easily upload it to the tool and go over to step number 4 to analyze that data. So, again, you could go through each of these individual traits, or you can get a sense of the overall, now looking both across the traits and across the different sites to get a sense of how varieties performed for how each of these performed across these sites. So taking a look at this, you can see this is front porch yield, and we have 4 replications here rather than, that's why there's 4 black dots. And again, looking at this, we can see that there's sort of a separation where, Lastic and Yacoro Rojo, just, stand out amongst the above, the low ranking, lower performing varieties, with Canis kind of floating around in the middle, straddling between, Alturas, Diva, Foisey, and Red 5, yielding lower. But you can see that there's certainly a fair degree of variation, replication to replication here for yield. You can look at like Lassic there and see, that ranged, from maybe something like 1,200, pounds to acre up to, 20, 400, wide range. And that's, something that you can kind of, visualize. And, one of the advantages of seeing the actual individual data points as well as the averages. This is, I often find that as of right now, that it's actually a little easier to look at these tables in the downloaded PDF version. So this is it in the downloaded PDF version. I apologize, there is a little error here where it says yield front porch and then yield front porch .1. It actually should be the first yield is actually the College of the Redwoods yield, and then the second one is the front porch yield. But this shows you again a way to look across. So thinking again where yellow is high and the purple, the dark purple is low. For all of these traits, high is good, except for this rust incidence where you actually want a low number. So you can kind of look across and see. All right, for yield, Lassic here standing out, though, Yacoro Rojo not far behind. For height, which was important for weed competitiveness, we see Foisey, standing up above everything else, literally. And, likewise, Foisey also had really good protein numbers. And you can see even that it's, for protein, if you look at that means grouping letter, it has, in College of the Redwoods, it's the only one with a mean in the means group A. That means nothing else was statistically close enough, was statistically close to it. Likewise, at the front porch farm, this protein was far superior to everything else. So this gives you a sense. Let's just look at rust here real quick as well. And you can see, in this case, low being good, Lassic had no rust incidents, though we couldn't say that was really statistically different than Alturas that had very little, or Diva that had 12%. Whereas Yacoro Rojo here had 72%. So which was only similar to Red 5. So going back to how do we make sense of this? Looking on what are the traits that are the most important for you? Is yield the most important? Well, looking at yield and resistance, then if prioritize those, then last bias coming out of this trial. If we are concerned about weed competitiveness and having to plant and having protein, then Boise's looking really good. Or maybe to cover your bases. So
just a bit. I'm sorry to interrupt you.
I'll wrap up by saying that though even in a trial like the wheat trial where we grew it in multiple locations, it's still a single year may not be reliable, especially if it's just you're doing trial in one location. So it's always wise to take a single year with a grain of salt, and if you can replicate trial a second year. That being said, it doesn't have to be perfectly repeated. You may want to eliminate some of ones that perform the worst that you don't need to look at again, and add some new things. As new things, you discover new things or new things become available from seed companies, and kind of continue to iterate and improve that trial. But certainly, it makes sense to not put too much weight on a trial that you just did in one location in 1 year. So with that, I would like to wrap up and turn it over for questions.
Okay, thanks, Jared. Let us move on to the Q&A here. And I also wanted to mention that this entire webinar and the one before it, if you missed it, are all part of the On-Farm Variety Trial Toolkit, which is available on this website. And there's a link there to the new Variety Trial Guide that was written by the Organic Seed Alliance very recently. And there's also a link to the tool that Jared explained. And there are also some other helpful data sheets there as well. So, okay, how large should a trial be for garden vegetables?
I can start to answer that question. This is Kit, and it really depends based on which vegetable you're interested in. And we have a great table in the variety trialing guide that Alice mentioned that you can find on the links that are up on the site right now. And in the guide, there it outlines for different families of crop how many individual plants you would need to observe in order to get a representative idea of how that crop performs in your environment. And so once you understand approximately how many plants you would need to evaluate in order to see how they're doing, then you can get a better idea of how big your trial needs to be. And it also would depend on your goals too. If it's something that you could evaluate given just a few plants, then you might be able to do a slightly smaller trial. But if you want to be a lot more specific and you want to really understand how a variety performs on average, then you might want to look at more plants. But I think Jared wants to get on that too.
Sure. So some quick, hopefully you can all hear me for a second here.
Yes, we can. Much better.
Okay, great.
Okay.
Okay, good. So like Kit said, there's a table in our variety trial publication, but a lot of that, there's lots of caveats, just like Kit said. So some of it depends on the crop, but it also will depend on the amount of variability you might expect to see in that crop. So something that's open pollinated and genetically variable, you're going to have more plant to plant variation than something that's either a self-pollinated crop like lettuce or a hybrid variety. And so those you would expect will be more uniform within a given variety, and you don't need as many plants. That being said, larger plots in general will be more robust to potential problems in the field, like a cultivation or a leak in the irrigation, or a gopher, or anything like that. If you've only got a couple of plants, especially if the trial is not replicated, then you put those plots at more risk the smaller they get.
Okay. They want to know about using statistical methods of analysis of variance and that sort of thing, whether you do that.
So looking at the trial tool that we have online, in essence, what we're doing is, in a way, this is looking at an analysis of variance, and that you're comparing the amount of variance within a given variety, across the replications, to the amount of variance between the varieties, to get a sense of, we're using a model that allows us to get a sense of how much variability there is. It doesn't, this tool is not actually, showing you a formal ANOVA, formal analysis of variance, to get a sense of if there is overall significant effects, across all varieties. That's something that would be relatively easy to add to the, add to this variety trial tool. In general, I would say, speaking for myself personally, and Julie, feel free to disagree with me, but in general, an analysis of variance is going to tell you less than whether or not something is showing whether or not something has is sharing a means grouping with something else, because that's reflecting the amount of variability that you're seeing, looking at on an entry by entry basis. So, for example, you could see basically a significant difference between entries in an analysis of variance, but that may just be because there's a single entry that is so very much worse than everything else. And so kind of the analysis of variance gives you kind of the first step in your statistical analysis, but it's maybe not giving you the information about whether or not which varieties are actually statistically superior, which is usually what you're more interested in.
Okay, someone wanted to know whether there's a recommended distance apart for the replications.
What I would say to that, and again, I welcome my other panelists to jump in here, is that you're actually, the closer you can keep the balancing act. So kind of going back to that question about how large should a trial be, how large should each individual plot be? There's a balancing act between having larger plots to both represent more of the variation that might be in a variety, and also to mitigate some of those field effects a little bit. But the larger your trial gets, the more distance between entries within a replication, and even the more distance between replications to a certain extent, the more variability you get in a trial. You'd like to have your trial be as uniform as possible, and especially if it's unreplicated. If it's replicated, you do have more wiggle room in terms of having distance between each of your complete blocks, if you're doing a randomized complete block design. But certainly within blocks, you would like to actually have your varieties as close to each other as possible, because then you're less likely to have the like you said, the climatic, soil, or other variability differing between one edge of your block and the other edge. So, like you said, it depends on the it depends on the amount of variability, and that, of course, is kind of the ultimate thing. Having a as uniform of a field as possible is going to be the first step in success. But then within that, if this push and pull of trying to keep your trial relatively compact, a square trial is better than a super long trial. Trying to minimize the distance, the actual feet between your entries, between your plots, is going to improve the performance of your trial. One of the things I point to is in those wheat trials that I showed as an example there. Initially, we had very long plots. We had 100-foot long plots, and what we realized is by going down from 100-foot long plots to 25-foot long plots, we're actually getting better results in our trial because we were able to have a more compact trial overall and a more field uniformity.
Okay, I just want to remind everyone that you can find the On-Farm Variety Trial Toolkit at the link on your screen, and that you can find the other webinar in this series as well as many other recordings about organic seed production and variety trials at the first link on our screen in our webinar archive, or you can go directly to the eOrganic YouTube channel and you'll find all those resources there. For example, from the Organic Seed Growers Conference, there's a playlist that has all of the recordings that we did at that conference, including one on variety trials. So with that, it doesn't look like we have any more questions, but I would like to thank our presenters today, Kit, Julie, and Jared, for sharing your information with us. And I'd like to thank everyone for joining us.